Hello students, in previous classes we are discussing about cell and their organelles, and in last lecture we discussed about endomembrane system and organelles of endomembrane system. Today we are going to discuss about another organelles. The first organelle is ribosome. The ribosome is firstly observed by Ch-8 in electron microscope, which is look like a dense particle and it is without membrane. So the ribosome is organelles without membrane, and it is composed of RNA, which stands for ribonucleic acid. And these ribosomes are two types. First one is 18S and another one is 70S. 80S ribosome is present in eukaryotic organism or eukaryotic cell, where 70S ribosomes are present in a prokaryotic cell and in a mitochondria and in chloroplast. This 80S ribosome is composed of two subunits. First one is a 60S plus 40S, and the 70S ribosome is composed of two units. First one is a 50S and another unit is 30S. Here this S stands for Sweetberg unit or sedimentation coefficient, which is indirectly suggests the density of the molecules, which also suggests the moving velocity or moving rate of these molecules in the centrifugation. And the main function of these ribosomes are protein synthesis, as we have seen earlier that in the endoplasmic reticulum there is one type of endoplasmic reticulum which is a rough type of endoplasmic reticulum. On the surface of this endoplasmic reticulum there are ribosomes present on that membrane of endoplasmic reticulum, which is responsible for the formation or synthesis of protein. So our next organelle is cytoskeleton. The word itself says everything, that cyto means cytoplasm and skeleton means we know what is skeleton. So the cytoskeleton is network like filamentous proteinous structure which is found in a cytoplasm of the cell known as cytoskeleton. Now what is the function of this cytoskeleton? Cytoskeleton gives support to the cell and their motility, and this cytoskeleton also maintain the shape and size of particular cell. So this is about cytoskeleton. Now the next organelle is cilia and flagella. First of all, cilia and flagella both are the outgrowth from the plasma membrane. This cilia and flagella is, you can see here, this is the cell and this is the flagella, which is outgrowth of plasma membrane, okay, and it helps in the movement of cell. Now let us understand about a flagella. Flagella is a larger organelle than the cilia, which is larger in size, and when we see in a electron microscope, we see in a structure like this. Suppose this is the flagella, and if we cut here and see in the microscope, so TS of flagella, in TS of flagella is shows that the outermost membrane of the flagella is made up of plasma membrane, and the core of this flagella known as axoneme. In the core, there is two microtubules present called central microtubules, which is connected to each other, and there is 9 peripheral microtubules, which is duplicate, and all these are connected with each other by the linker. And this central part of the flagella known as axoneme, which is covered by the one sheath-like structure. So this is the ultrastructure of flagella. Now what is the role of flagella? Flagella is very useful in the movement of cell in a given direction or particular direction, which is emerge from the basal body like centriole, and this flagella is emerged from the centriole. The portion of proportion of microtubules in a flagella in a 9+2, where 9 is a peripheral microtubules and 2 is a central microtubules. So this is about cilia and flagella. Now the next organelles is centriole and nucleus. So let us see about centriole and nucleus. The another organelles, centrosome. The centrosome is made up of two cylindrical structure known as centriole, which is arranged in manner of perpendicular to each other. Means the centrioles are arranged in perpendicular to each other and it form centrosome. Each has organization like cartwheel. You can see here, this is the structure of centrosome. It forms cartwheel like structure here. It made up of 9 peripheral fibrils of tubulin protein. Means it made up of 9 peripheral fibrils. You can see here, these are the 9 peripheral fibrils of tubulin protein. Means these are the tubulin proteins, and it is present in number 9, where in the central part unlike flagella, they don't have central or tubulin or fibrillin protein or microtubule protein. This central portion is known as hub, and this central portion hub is joined by or connected with the peripheral fibrils by the structure known as radial spoke. Are you following? The central portion of centriole known as hub, and it is connected with peripheral fibrils by the structure known as radial spoke. Okay. Now what is the function of this centrosome? The centrosome is actually present in only animal cell, as we all know, and the main function of centrosome is spindle formation during cell division. It means centrosome is responsible for the formation of spindle formation during cell division. Now one question may come arise that it is okay that in animal cell this centrosome is responsible for the spindle formation, but in the case of plant cell, from where this spindle fiber or spindle formation take place? So this is the question for you, and you have to find out this answer of this question, that from which portion or which organelles this spindle formation occur in a plant cell. We know that in animal cell spindle formation occur in due to centrosome. Okay, so main function of the spindle formation, main function of the centrosome is to spindle formation. The structure of centrosome is having 9 peripheral fibrils made up of tubulin proteins, and the core of this centrosome known as hub, which is connected with the peripheral fibrils of tubulin protein by the radial spoke. Okay, so this is about centrosome. Now next organelle is nucleus. We all know that one of the most important organelles of cell is nucleus, but there are some cells which don't have nucleus. For example, our RBC, red blood cell, don't have nucleus. In plant also there are some cells present like sieve tube and vessels don't have a nucleus. Now let us see about nucleus. After Robert Hooke, one another scientist, Robert Brown, who discovered the densely stained region in the cell, and later they given the name of this organelles as a nucleus. Then after Robert Brown discovery of nucleus, one another scientist come. The name of the scientist is Fleming, who discovered the material present in the nucleus called as a chromatin, which is stained by basic dye, and the name of that organelles or present in the nucleus given that name of a chromatin. Means it is stained by basic dye and found in nucleus, which is network like structure of nucleoprotein. The chromatin is highly extended and elaborated nucleoprotein fibers called as chromatin. So means what is chromatin? So chromatin is highly extended and elaborated nucleoprotein fiber present in a nucleus known as chromatin. Now this nucleus when we study in a electron microscope, it shows that it have double membrane structure, outer membrane and inner membrane, and in outer membrane and inner membrane there are space present between them, and this space known as perinuclear space. The space here between inner membrane and outer membrane, the space present, and the space of this known as perinuclear space. This outer membrane is sometime or it is continuously with an endoplasmic reticulum. We know that, and this endoplasmic reticulum have sometimes ribosome also, which function of the function of this ribosome containing endoplasmic reticulum is protein synthesis, and the endoplasmic reticulum without ribosome, the function is a lipid synthesis, inner lipid synthesis. Sometimes this nuclear membrane or nuclear envelope having rupture in them and having pore-like structure, and this pore known as nuclear pore. This pore is responsible for the movement of DNA, RNA and protein from the nucleus to cytoplasm and cytoplasm to nucleus. Then another organelle present in nucleus is a nucleolus, which is highly dense organelles found in a nucleus without membrane. Remember, my friends, that this nucleolus don't have any membrane surrounded them, and the main function of this nucleus is to produce ribosomal RNA, and this nucleolus is highly abundant in the cells, which is function in the protein synthesis. Now, during interphase, this nucleus shows having chromatin-like structure, but when this cell goes into the cell division, this nucleus and chromatin disappear, and this chromatin forms the structure like this, known as chromosome. These chromosomes only present in cell during cell division, the phase of cell division. But when cell is in interphase, they shows they having chromatin. This chromatin have DNA and protein. The protein is histone protein. The DNA is packaged in the histone protein that will be discussed in detail in the class 12. Now, the DNA present in one cell around 2 meter long, which means the 2 meter long DNA is packaged in the one nucleus or one cell. Just imagine that 2 meter long DNA is packaged in that way that वो इतनी जगह में पैकेज हो जाता है कि हम जानते हैं कि सेल की साइज कितनी है, उसमें भी उस तरह से वो डीएनए पैकेज होता है कि उसमें स्मॉल जगह में भी वो पूरा डीएनए 2 मीटर का डीएनए पैक हो जाता है. ओके, आफ्टर इंटरफेस व्हेन सेल एंटर इन द सेल डिवीजन, the nucleus and nucleus disappear, and the chromatins form the structure like this, known as chromosome. The chromosome having central portion known as centromere. It also known as primary construction. This centromere having one another structure near them. It is a kinetochore which is present near the centromere, and on the basis of this centromere, chromosomes are divided in a four type. First one is a metacentric chromosome. When the centromere present in a middle of the chromosome, that type of chromosome known as metacentric chromosome. The another type is submetacentric, when the centromere present slightly away from the middle portion of chromosome. This type of chromosome known as submetacentric chromosome. Then acrocentric chromosome. In acrocentric chromosome, the centromere present closer to the end. You can see here that centromere present near the end of the chromosome. And last one is a telocentric. In telocentric chromosome, the centromere is present at the end. Okay, so this is about nucleus. In today's class, we discussed about centriole, nucleus, ribosome, and cytoskeleton. Here we completed our chapter, Cell, the Unit of Life, in next class we will start another chapter. So thank you students.